Okay. As I said, this chapter is about being able to study a habitat or study an ecosystem. So, what I will be doing now for the next few minutes or so is just taking you through how, in theory, would we conduct an ecosystem study, or within an ecosystem itself, maybe look at the individual habitats. So, first of all, please go away and learn those definitions. So, an ecosystem, remember, is a large thing. We're talking about organisms and their interactions in a physical environment. Okay. So that could be something like, for example, a grassland. It could be something like, for example, a woodland. It could be a river, or it could be a coastal region. But a habitat is much smaller. You're talking about the specific place an organism lives. So the ecosystem that we'll be studying is a grassland ecosystem. And I want you to bear in mind what kind of organisms would you expect to live in a grassland in Ireland? Okay, so we have to look at this in the Irish context. Um, in relation to an ecosystem, the ecosystem we'll be studying is just a grassland out in the park. Okay, and if you think about the dominant species there, obviously the dominant species is going to be grass. Okay, so grass is dominant, but you must ask yourself the question: how is grass so dominant in these grasslands? Okay, what management processes here are involved here by humans that are maybe encouraging grass, and maybe what adaptations does grass have to make sure that it is the dominant species in the grassland? Now, grass is not the only species, of course, that's surviving here in our grassland. There are many other organisms as well. So you can read a lot of this in your own time, but I'm going to look here at the step three here, whereas we're looking at the abiotic factors in the habitat. Okay, so I'm going to take the lot of you out, hopefully sooner rather than later. And we're going to go out and we'll have a look at the grassland, and we're going to look at the abiotic factors. Remember, abiotic factors are non-living factors. Okay, so non-living factors, remember, might include air temperature. Okay, so temperature around us in Ireland right now, it's absolutely Baltic. So it's probably temperatures outside, maybe 6, 7 degrees air temperature. Um, but when we go out, it'll be probably on average might be 15, 16 degrees, hopefully. Okay. The soil temperatures will have a huge bearing on organisms that can live in that physical environment. Okay. So, like, I mean, if you think about it, soil temperatures, if the soil temperatures are below freezing, um, then very difficult for organisms like worms and different things to kind of live, and do their normal processes. So soil temperature is important. It's important for the plants as well because grass needs a certain temperature really to kind of thrive. Okay, and the mild temperatures of Ireland year round tend to do the grass favors. Soil pH. So pH, remember, is basically how acidic or basic something is. And for soil pH, what you would do there is you would literally stick a pH meter or a sensor into the ground, get an indication. Now, oftentimes we would find that soil is acidic in Ireland. So what people do, and what farmers do oftentimes, is they spread something called lime, and lime will change the pH and make it more basic. But the ideal pH for grass is a pH of around 6, 6.5. So that would be something to bear in mind. And light intensity then as well. So light intensity, we will have a light meter for that. And what we will do is we will measure how much actual sunlight is getting through down to the I suppose organisms we're concerned with, which is the plants that are living in the grassland. Okay, so we'll expose the light, and one precaution there that you have to do there is to make sure that you don't cover it with your shadow so that you get an actual true indication of the light levels. So the different, I suppose, apparatus that's required: a thermometer, of course, for temperature. As we said, you can use a pH sensor, or you could use universal indicator. But if you're using universal indicator, we need to use a color chart, remember? And we have something called a light meter for light intensity. So that's pretty straightforward there to remember. Right. So once we have got an idea of the abiotic factors, okay, the non-living factors, the next thing you do then is you actually try to identify some organisms in the habitat. Okay, so we're going to have something called a key. Now, a key is, I suppose, I don't know, have we seen one of these since first year actually, but basically what it is a series of questions that help you identify the species in front of you. Okay, so for example, here we have an example here for trying to identify a tree. Okay, and it says here, are the leaves single or are they divided into leaflets? So tiny little leaflets. Is it just a single leaf or leaflets? So if it is leaflets, then so if it's single, you go to number two. Okay, but if it is leaflets, you go to number four. So I'm going to go to number two here. Are the leaves divided into many lobes? So the rounded parts here. So again, if it is lobes, then it is an oak, or if it's not divided into lobes, you go to number three. So you probably get the idea there. It's basically a series of questions. It's almost like a treasure map. You follow the treasure map until you find the organism in front of you. Okay, and we use this to identify different organisms. Now, you might think that this is very easy to identify different organisms, but actually, um, especially I suppose for ecologists and their daily work, it's very specialized, and many of these I suppose whether it's plants or animals look very very similar, that it really sometimes takes experts to identify the difference between them and identify different species. Right, so what we need to do then is we need to think about, I'm going to skip on here, we need to think about catching various animals and maybe looking at different plants as well. So there's different equipment that you would require to do this. So in the grassland, what we're going to try to do is we're going to take out something called a pooter. Now, I may have introduced a pooter to you before. A pooter is used to collect small insects in the grassland. Okay, so what we would do is we would look at the leaves of plants, or maybe look closer to the, I suppose, plants on the ground, and we would see, do we see any crawling insects on it? And if we do, maybe we want to use a pooter to try to capture that organism. So what you would do is you would put one end of the tube over the insect, and you would then create a suction by sucking through the other end of the tube. Okay, and but that suction then draws the insect in, and you can have a look at it here inside your pooter. You then use your key. So your key is used to identify the organism, and you ask yourself the questions again: can I identify it? So in Ireland, the kind of things we'd expect to find in grassland, well, there's lots of different things. Obviously, you could even find things like ladybirds. You could find things like spiders, green flies, and other kind of insects. So if it's an unknown insect, you need to use your key to find out what it is. Right, another thing then is we use something called a beating tray. So we'll be taking out all of this again, as I said. A beating tray is effectively in the grassland. There might be some bushes that might just happen to be in the grassland that we're looking at. And what we'll do then is we'll see, okay, is there any of the organisms different on this bush than the general grass? Okay, so what you would do is you'd shake the bush, and you would then the insects would fall onto. You could use a sheet or you could use a tray, and whatever insects fall into the tray, you would then again try to identify them using your key. The pitfall trap. The pitfall trap is designed to capture crawling insects, maybe that you mightn't see at first because they live so close to the soil. So what you do is you dig a hole, and in the hole you could add a cup or you could add a jar, and you just leave it for I suppose a bit of time, ideally a few hours. You come back, you check it, and what you should find there is that insects have fallen into the tray or into the pitfall trap, and you can identify them then. So this would be things that you would expect to fall in would be things like ants, for example, would fall into your trap. Okay, right. So then the next thing is you want to actually study these organisms. And now that I have an idea of the different types of organisms in the habitat, I might want to test, um, or figure out how are these organisms so well adapted for their environment. Okay, now you'd have to do a little bit of research on this. So say for example, I find, okay, grass is the most numerous type of plant we find in a grassland, and the particular type of grass you might be talking about there is something called perennial ryegrass, something that we use to feed animals. Okay, we use it to feed cows. And the reason that the perennial ryegrass is so common is because it is adapted to high light intensity conditions. Because if you think about it, there is no shade in a grassland. Light, the light will be very high intensity in the open grassland, and not all organisms can cope with that. So grass is adapted to survive and thrive in very high light intensity conditions. Other organisms that we might find would be inside there, and their adaptation might be something, for example, buttercups. Okay, buttercups have bright yellow petals, as you know, to attract pollinators. That's an adaptation. Okay, I suppose there's other things as well. Oh, can't think of the name of them now, but you're talking about the sticky kind of plants that basically will add their seeds to your clothes, and you carry it away, and that's an adaptation of that plant to basically spread its seeds around the place. Okay. An animal adaptation, well, we know that earthworms are dark because that matches the color of the soil, and therefore things like birds find them harder to find, okay, because their vision mightn't be as good. Okay, so that is your ecosystem study in terms of the theory so far. And there's other things here then I must come back and look at. Um, but there's other things here as well. I don't know, does it mention it here, which is yes, it does, is a quadrat survey. So this is one of the this is one of the fundamental things we will absolutely do here to see which, I suppose, organisms are, and we can use something called the DAFOR scale, which is D for dominant, A for abundant, F for frequent, O for occasional, and R for rare. And what we will do is we will take out these quadrats with us. The quadrats are normally grids, and they would have little squares in them. You've probably seen them inside in the lab before. And what we will do is we will go out to our grassland, and we will throw a peg over our shoulder to make it nice and random. In our grassland, we will place the quadrat down, and when we observe, I suppose, species, we'll observe the species in present within the quadrat, and to identify what's, again, as I said, dominant, abundant, frequent, rare, or occasional. So I will explain this in detail when we do this, but just be aware of this. This is used to, I suppose, determine the frequency of different organisms. And what you would do is you'd repeat the survey 10 times, and you get a percentage out of 100. And this would tell you then, again, as I said, how frequent it occurs within your ecosystem. Right, so that's kind of just an introduction to the different methods we use inside in an ecosystem study. And we will do it physically then, hopefully, as soon as possible.